Asymmetry of lipid bilayer. The phospholipids in plasma membranes are asymmetrically distributed across the bilayer. The amine containing phospholipids are enriched on the cytoplasmic surface of the plasma membrane, while the choline containing and sphingolipids are enriched on the outer surface. This asymmetry is usually not absolute except for glycolipids. In the human erythrocyte, for example, the phospholipids such as sphingomyelin and phosphatidylcholine are mostly found in the extracytoplasmic leaflet, whereas phosphatidylserine and phosphatidylethanolamine are preferentially located on the cytoplasmic face. The maintenance of transbilayer lipid asymmetry is essential for normal membrane function. Once lipid asymmetry has been established, it is maintained by a combination of slow transbilayer diffusion, protein lipid interactions, and protein mediated transport. Role of lipase and flopases. Although the barrier to rapid spontaneous flip flop contributes to the maintenance of lipid asymmetry, other mechanisms must be responsible for the regeneration of lipid asymmetry. Perhaps the most significant contributors to the maintenance and dissipation of transbilayer lipid asymmetry are proteins that catalyze the movement of the lipids across the membrane. These activities are classified according to substrate specificity, requirements of energy, and direction of transport. Two classes of transport activities have been described that are responsible for the ATP dependent transport of lipid. The most well characterized activity catalyzed by flippase, which is a P type ATPase, transports glycerophospholipid from the outer monolayer to the cytoplasmic surface of the plasma membrane. A second ATP dependent activity catalyzed by floppase, which is a ABC transport protein, transports lipid in the opposite direction. The plasma membrane also contains phospholipid translocator termed scramblase, which ensures that both monolayers remain equally populated with phospholipids. It moves phospholipid non-specifically in either direction along the concentration gradient. It does not require ATP but is activated by calcium. Fluidity of a lipid bilayer. The fluidity of the lipid bilayer, the optimal value of which is a prerequisite for normal cell growth and function, depends on lipid composition and temperature. In general, an increase in the temperature tends to elevate membrane fluidity. Lowering of temperature causes a freely flowing, low viscous, fluid like membrane structure to a more rigid gel like organization. This change is described as phase transition, and the temperature at which this happens is called transition temperature. Fluidity of lipid bilayer also depends on chain length and the degree of unsaturations of its component fatty acid residues. Cholesterol is also a major determinant of membrane fluid. At high temperature, cholesterol interferes with the movement of the phospholipid fatty acid chains, making the membrane less fluid, but at low temperature, it interferes with interactions between fatty acid chains and prevents membranes from freezing. At high concentration found in eukaryotic plasma membranes, cholesterols tend to make the membrane overall less fluid at growth temperatures near 37 degrees Celsius.